In this video, we will study about bacterial viruses. Bacterial viruses are viruses which parasitize bacteria. They are also called as bacteriophages. Now, first we'll see certain general characters of viruses. Viruses, they are very small in size, so they are ultra microscopic. They are not made up of cell, non-cellular, and they cause many infections, so called as highly infectious. They can infect plants, animals, human beings, as well as bacteria. Viruses can only multiply inside the living organism, and outside the living organism they are inert. We can store them in the form of chemicals. They are obligate parasites. Now, this term was coined by Louis Pasteur. Obligate means they will only live with the living organisms, so they can infect plants, animals, bacteria, and human beings. And viruses which parasitize bacteria or infect bacteria, they are called as bacteriophages. So, in this video, we'll study in detail about bacteriophages, also called as bacterial viruses. Best studied example is of Escherichia coli, coliphage. We also call it as coliphage. If we see structure, it is tadpole-like. And viruses are also called as super parasite because they take control of host machinery all over. They behave like Hitler, we can say. Now we'll discuss about structure of bacteriophage. Now, if we see the structure of bacteriophage, we can see in this diagram. It is tadpole-like. Now, this part of the virus is head, and if we cut its section, so we can say this part is head, which is hexagonal and prism-like. Inside, this encloses genetic material, and this is DNA, which is double-stranded. And this part is called as its tail. This is neck and collar, and this tail is made up of a sheath. In the center is present a core called as tail core, and here hexagonal end plate is present. This part is hexagonal, which is having 6 spikes. 4 are visible, 2 will be on the back side, and to them are attached fibers, and these are called as tail fibers, tail pins. So this is about structure of tadpole virus. Now we'll again study. So this is tadpole-like virus. It is hexagonal or prism shape in outline. This outermost coat is actually made up of protein, so this is called as protein coat. And inside is present double-stranded DNA. Now this tail sheath, which is elongated part, this part is contractile in nature. This has hexagonal head, hexagonal end plate, which bears 6 spikes to which are attached 6 fibers. Now we'll discuss about life cycle of bacteriophage. This was all about structure of the bacteriophage. Now what happens when infection occurs on the bacteria? Now this phage will get attached. Now supposing this is bacteria and this is the virus. Now this will get attached with the tail fibers to it. Right? This will get attached like this. So tail fibers will help in its attachment. Now this part of the bacteria, now this will be dissolved with the help of enzyme lysozymes, right? And when this is dissolved, we can say a hole will be created in the bacteria, or a hole is created. So we can see here hole is created, right? Now this tail will contract, right? When this contracts, this will become shorter, and DNA which is present over here, now this will get injected into the bacteria. So this DNA will be injected into the bacteria. Now empty protein coat will be left behind. This is also called as host. Later on this will get degenerated. Now latent phase, next phase is latent phase. This is infection phase. Now in the latent phase, what happens actually? Now this is the viral DNA. This is the nuclear DNA of the bacteria. Nucleoid, we can say. This is not present in the nucleus. Now this viral DNA will take control of whole of the bacteria, right? And this will start synthesizing enzyme nucleases. Nucleases are enzymes which can cut this DNA into pieces, right? So these nucleases will digest this nucleoid or bacterial DNA into pieces, and their pieces will be produced. Now meanwhile, what will happen? DNA will start forming viral DNA and viral protein particles, because Viral DNA take control of the host machinery. So bacteria will not form anything for bacteria itself. This will act as a slave, and this will start forming viral DNA and viral protein particle, and this is called as vegetative phase. So replication of viral DNA will also take place. Formation of viral protein will take place. And ultimately, when lot of viral protein and viral DNA particle have been produced, and these are the components, so we can say machinery of the virus, right? A tail part. So ultimately, they will assemble. Like supposing this is the protein code and this is the DNA. Now this will enter into the viral DNA, as well as this part will join, tail part will join the viral, and ultimately complete viral particle is produced. So this is called as assemblage of viral DNA and viral protein to form complete phage particle. So this is the vegetative phase, and we can see here also these are phages which have been created. The next stage is when about 100 to 150 bacteriophages have been produced inside the bacteria. So this will put a pressure on the bacterial wall. This part will break up, and these phages will be released, and they will come out of this bacteria after their death. So phages will be released, and this causes lysis of the bacterial cell. So this is about, we can again repeat what are the different phases in the lytic life cycle. First is the infection stage, where virus will parasitize the bacteria, will get attached, produce the enzyme lysozyme. Lysozyme will dissolve the bacterial cell wall, and when this viral DNA will be injected into the bacteria. Now this viral DNA will take control of whole of the bacterial cell. This will degrade this DNA of the bacteria with the help of nucleases. Now bacterial DNA pieces will be created, and this will act like a slave, and viral DNA will replicate, viral protein particle will be produced, and viral we can say phage particle, they will get manufactured, and there will be then assemblage of all the viral particles, tail, head coat, and DNA, tail fibers. So ultimately viral particles are produced. When about 100 to 150 are produced, they will break the DNA, they will break the bacterial cell wall, and they will be released. Now they are ready to again infect other bacteria. So this is all about structure of bacteriophage as well as its lytic life cycle. Thank you for watching my video. If you like my video, please like, share, and subscribe.